Hello, my name is Maryam, and for my summer research project, I worked under Dr. Janice investigating a strain of bacterium called Comatigaebacter xylinus. I'll call it KX from here out. This bacterium is a major producer of bacterial cellulose and biofilms. If you're not familiar with the term biofilms, they are bacterial communities held together by self-produced exopolysaccharides, so sugars, like bacterial cellulose. Our understanding of bacterial cellulose and biofilm production is quite limited, and getting a better understanding of both of these processes has a broad range of potential applications. For example, the resilient nature of biofilms pose significant challenges in healthcare and agriculture. Understanding cellulose production, which is actually the focus of our lab, has a large range of applications as well, with some particular fields being sustainability, manufacturing, food, biomedical, and cosmetics. For example, bacterial cellulose has strong potential in manufacturing materials like paper, food packaging, and medical products, all of which would be biodegradable. So getting back to the research, KX, the bacterium we work with, is actually considered the highest producer of bacterial cellulose. You can see here, this is a liquid culture of the wild type, and just after a couple days, it's produced this much cellulose. So in my line of research, I worked with two strains, the naturally occurring wild type and a mutant called SDIA. This mutant strain is basically just missing a gene from its genome that is called SDIA. So this mutant typically does not produce cellulose. And in my fourth year thesis, I was actually able to find some evidence to suggest that the SDIA gene is involved with a bacterial communication process that typically leads to cellulose production. To put it simply, the mutant, which remember does not have the gene, did not seem to release these chemical words, so to speak, that the wild type very much did release. And so during this term, I actually conducted a procedure called complementation that I did on the mutant. Basically, I have been trying to reintroduce the SDIA gene into the mutant, theoretically making the wild type phenotype, which could then prove that it is in fact the SDIA gene that is responsible for the difference that I was seeing. This here is actually a gel electrophoresis showing the SDIA gene that I successfully joined into a vector before I transferred it into the mutant. The second project I carried out this term was investigating the effect of phytohormones on growth and cellulose production of the wild type and mutant. Phytohormones are plant chemical messengers, some of which have been shown to promote the growth of certain bacterial strains. The results varied greatly. Here I actually have my graphs for the effect of salicylic acid at different concentrations on the growth of the wild type and mutant strains, as recorded by optical density over time in hours. You can see there's a bit of difference between the mutant and wild type, which suggests that the SDIA gene may have a role in phytohormone response as well. Overall, the purpose of this research is to achieve a better understanding of the genetic and environmental processes involved in KX growth and cellulose production so that we may use it for innovative and sustainable applications in various industries.